this lecture, we're going to take a look at the mechanisms then that non-steroid hormones use to exert control on the activity of a cell. And then we'll look at overall control of endocrine activity. We'll answer the question of what is it that tells an endocrine gland when it's time to secrete. So non-steroid hormones are those, again, that are built from amino acids, a single amino acid, short chains or very long chains of amino acids. And they include hormones like epinephrine, also known as adrenaline. Epinephrine is produced by the adrenal medulla. The two thyroid hormones that are produced at the thyroid gland, growth hormone and oxytocin that are coming from the pituitary gland, and there are many more as well. And they work through a process known as signal transduction because they are unable to make it directly across the cell membrane the way that a steroid hormone does. They have to bind a receptor outside the cell, which then initiates activities inside the cell. Most of the non-steroid hormones aren't allowed into the cell. That's not true of all of them, but most of them stay outside the cell. And I think of them as ringing a doorbell so that they can be heard inside of the cell. So let's look at how signal transduction works in a large scale, and then we'll break it down into steps like we did with the steroid hormone. So here we have a non-steroid hormone that's found, and by the way, because they do dissolve readily in water, most non-steroid hormones don't require a transport protein. But again, some of them may need a transport protein to make it around in body fluids, but most of them just dissolve in the body fluids. So we have here then, so here's our steroid hormone, and here is an example of our protein that is going to transfer the signal inside the cell, do signal transduction. So we can think of, by the way, we can think of this hormone as a first messenger, as a messenger, the primary messenger that gets sent out into the body from the endocrine gland that secretes it. So this transmembrane protein found here at the surface of the cell has on the exterior a binding site. This is a place where the non-steroid hormone can bind to this receptor protein. Inside of the cell, there's what's termed the activity site. This is a site that will change its shape when the non-steroid hormone binds there at the binding site. And associated with the activity site is a second protein called the G protein. Okay, so let's see what happens then. Oh, and nearby, we should go ahead and say, nearby to this receptor protein and G protein is a third protein. It's an enzyme named adenylate cyclase. So this is a multi-step process, so we'll just take it nice and slow as we talk about it over the next couple of slides. So our non-steroid hormone binds at the binding site. This causes a change at the activity site. When that non-steroid hormone binds, it scoots electrons around in this molecule, and as those electrons move, then that changes the shape of the activity site. And as we bump all those electrons around there at the activity site, that G protein that's associated with the activity site, its electrons are going to be bumped around as well. And so this creates then an activated, I think of it kind of as an activated protein complex here. And when we activate the complex, then that will also activate a nearby enzyme, the adenylate cyclase. And what adenylate cyclase is capable of doing is taking our old buddy ATP, that's adenosine triphosphate, and converts it, rips off a couple of the phosphate groups, and, sorry about that, converts it into a version of this ATP called cyclic AMP. This time now it's adenosine monophosphate, and it's got a little circular structure associated with it. So we call that cyclic AMP, or CAMP for short. The CAMP now acts as a little, this is what we term the second messenger. So our second messenger can now run around inside the cell, getting things activated. And I keep touching the screen in a wrong way. So cyclic AMP, I think of it almost as the little yippy yappy dog inside the house. We ring the doorbell out here, and that gets the little yippy yappy dog running around looking for the master to make something happen inside the house. So the master in this case is a set of enzymes that are thought of as, or that are termed the protein kinases, and it's these activated protein kinases then, when we activate them with CAMP, that then are capable of producing whatever the cellular change is. The protein kinase will now move through the cell, activating processes that were inactive before, or deactivating processes that had been active. So here are the steps then. We see the receptor protein with its binding site outside the cell and an activity site inside the cell. The hormone binds, changes the activity site, and then we see the G protein activated, which brings the signal inside the cell. Once we get that activity site activated and the G protein is activated, then adenylate cyclase is also activated, allowing us to produce cyclic adenosine monophosphate out of our old buddy ATP. And now we have a second messenger inside the cell. Now cyclic AMP is not the only second messenger inside the cell. It's not the only yippy yappy dog that we can make, but it is one of them. So again, the G protein has to be activated in order for us to be able to get things started. So when the activity site is activated, the G protein becomes activated. The activated G protein then gets the adenylate cyclase all excited and working to make ATP into CAMP. And then CAMP, our yippy yappy dog, runs around inside the cell, activating protein kinases. These are enzymes that, when they are activated, can transfer from ATP that's found inside the cell, a phosphate group, to other proteins. So once we phosphorylate or add these phosphate groups to the proteins, then those proteins that have been phosphorylated are going to bring about whatever change we associate with that non-steroid hormone. Whew, I know that's a lot of steps. And so if you notice there in the Blackboard site, I have given you an extra credit assignment that has you drawing, or kind of outlining with a drawing or cartoon, how steroid hormones and non-steroid hormones do their thing. And you can earn up to 6 extra points for doing those two sets of drawings, if you wish. So what do we see in response to getting protein kinases activated in the cells? Well, we see that we can change the permeability of a membrane so that we can bring materials into the cell. We can activate enzymes. We can synthesize particular proteins that are going to be important to cell processes. We can cause other we can stimulate or inhibit, stop metabolic processes inside the cell. We can cause the secretion of other hormones or other hormone-like or materials, or even other cell products. For instance, oxytocin helps to initiate the delivery of milk from the cells that are making the milk proteins and sugars and fats, and we see secretion of milk from the mammary glands in response to oxytocin, and it can cause cells to start moving. So we can see cellular movement, cilia moving, or materials moving within the cell. Okay, so lots and lots of effects. So what other, I mentioned that there are other second messengers, and so But first, which of the non-steroid hormones use cyclic AMP as their second messenger? Here are the list, and these are the abbreviations that you're going to learn in the next chapter or next lectures. But TSH is thyroid stimulating hormone. ACTH is adrenocorticotropic hormone. FSH is follicle stimulating hormone. We've already talked about that one. LH, luteinizing hormone. ADH, we've talked about it too, antidiuretic hormone. PTH, you've heard of that one, parathyroid hormone. Epi is epinephrine. Norepi is norepinephrine. CT, that's calcitonin, you've heard of that one. And glucagon, these are all hormones that use cyclic AMP as their second messenger. But they're not the only second messenger. Diacylglycerol is a second messenger. Calcium ions can be a second messenger. They combine with calmodulin inside the cell rather than activating protein kinases. But when calmodulin gets activated with calcium ions, we can see a response. And frequently, inositol triphosphate is the chemical that causes the release of calcium ions from their internal storage locations. And then a second kind of nucleic acid, guanine, can be used to produce cyclic guanosine monophosphate, cyclic GMP. So adenosine monophosphate and guanosine monophosphate are cousins kind of molecules similar to each other. So lots and lots of materials can serve as second messengers. Prostaglandins, we talked about them very briefly, but prostaglandins are non-hormone biochemicals that also provide local control of cells, and they do it in mechanisms similar to those of hormones. But because they are very quickly deactivated once they're released out into body fluids, they only work locally. So they are a paracrine secretion, these prostaglandins are. And we've talked about prostaglandins already as well, about how those prostaglandins that are part of semen, for instance, can help to trigger smooth muscle contractions in the female reproductive system. So what are some effects of prostaglandins? We see relaxation of the smooth muscle that's in the airways of the lungs and in blood vessels. So they're at capillaries or small arterioles. We can see relaxation of the smooth muscle surrounding the blood vessels. We see contraction of smooth muscle, for instance, at the uterus. This can stimulate local cells to release hormones, these prostaglandins can, and they might promote inflammation. So lots of activities that different prostaglandins can initiate. Alright, so the last piece that this lecture is going to involve, or is involved in this lecture, is how is it that we let a hormone, or let an endocrine gland know that it's time to release the hormone that the gland has produced. And hormonal secretion is almost always controlled by negative feedback. So we have a signal that causes the hormone to be released. For instance, low calcium in the blood is going to trigger the release of parathyroid hormone. So as we release parathyroid hormone, then we stimulate osteoclasts to break down calcium, and we do other things as well, but let's just focus on that one. So osteoclasts break down the calcium. This causes an increase in blood calcium, and that's going to then reduce the call for parathyroid hormone. And as we have less parathyroid hormone out there, then calcium levels in the blood may decrease once more, which will then call for more parathyroid hormones to be released. So remember negative feedback, where if this is homeostasis, as we move away from homeostasis, activity will be initiated that pulls us back toward homeostasis. Out of homeostasis again. We change activity, we go back into homeostasis. That's negative feedback. So how do we get rid of the hormone that's been released? Well, there are lots of ways. We can break it down at the kidney, or we can excrete them at the, break them down at the liver, or excrete them at the kidney. Those are big ways that we do it. And so we can talk about half-lives for hormone. And so the half-life is the amount of time it takes for the concentration of the hormone to be reduced by 50%. So if we have two parts per million hormones in the blood, then the half-life is the amount of time that it takes to get the concentration down to just 1 part per million. So hormones that have very short half-lives tend to act quickly and then be deactivated. So epinephrine and norepinephrine have relatively short half-lives, on the order of minutes. The half-life of epinephrine and norepinephrine is. And so when we release hormones into the body, there are processes that are going to be working to remove and shut it down. Hormones that have really long half-lives, though, so for instance thyroid hormone and many of the steroid hormones have very long half-lives, on the order of days, can have then effects that can last for days and even longer after they're initially released into the body. So the shorter the half-life, the more rapid the effect, and the more rapidly the effect is shut down. So what are some specific control mechanisms? So it could be that the hormone is released when another hormone binds at the cells of that endocrine gland. So for instance, we talk about these, by the way, as the tropic or regulatory hormones of the body. So a tropic hormone is one that has an effect of causing the release of another hormone. And so the tropic hormones or regulatory hormones of the hypothalamus are very important in stimulating or inhibiting, because we have inhibiting and releasing hormones that are produced at the hypothalamus, and they cause other hormones to be released. So for instance, let's just see it for instance here. We see the hypothalamus here. The hypothalamus produces a hormone called thyroid releasing hormone. Thyroid releasing hormone acts at the anterior pituitary gland and causes the release of thyroid stimulating hormone. So thyroid releasing hormone is a tropic hormone, acts at the pituitary and forces the pituitary to release thyroid stimulating hormone. Thyroid stimulating hormone is also a tropic hormone, and it acts at the thyroid, causing the thyroid to release the two thyroid hormones, T3 and T4. We'll talk about the names of those later on. So two tropic hormones are required to get T3 and T4 released. T3 and T4 are not tropic hormones. Their job is to go out, talk to all the cells in the body, and cause them to grow more quickly and divide more rapidly. Okay, so one way that we can control an endocrine gland secretion is with another hormone. A second way is through direct neural stimulation. This is how we get epinephrine and norepinephrine released in the body under emergency situations. So if you're driving down the highway and somebody tries to pull into the lane and into you in the process, you lay on the horn, beep, and you get, you find yourself a little breathing heavy, feeling really nervous and anxious. All of that is because the sympathetic division of the autonomic nervous system has directed your adrenal glands to release epinephrine and norepinephrine. The third way that we can control the release of hormones is by changes in the composition of the internal environment of the body. So for instance, as blood glucose goes up, the pancreas releases insulin. As blood glucose falls, the pancreas secretes glucagon. So we've talked about that process before as well. And And so these are negative feedback mechanisms then that work to keep blood levels of the hormone relatively constant so that we are maintaining the body in homeostasis. So there you have it, all the functions of, or the mechanisms through which non-steroid hormones have their effect in cells using second messengers, and the ways that we turn off and turn on the secretion of hormones from endocrine glands. All right, you know what to do. If you have any questions, don't hesitate to reach out to me.